Translation. RNA to protein definition. Translation is the biological process by which the sequence of nucleotides in mRNA is decoded to build a chain of amino acids, resulting in a functional protein. It is the second major step in gene expression, following transcription. Where it happens: prokaryotes, cytoplasm, no nucleus. Translation begins during transcription. Eukaryotes, cytoplasm, specifically on ribosomes, free or attached to rough key players in translation. Component function. mRNA carries the genetic code from DNA. RNA brings specific amino acids to the ribosome. The structural and catalytic part of ribosomes. Ribosome site of protein synthesis, made of two subunits, large and small codon, a set of 3 mRNA bases that codes for one amino acid. Anticodon, a set of 3 bases on RNA complementary to the mRNA codon. Start codon, AUG, signals the beginning of translation. Codes for methionine. Stop codons, UR, WAG, UGA signal the end of translation. Phases of translation. 1. Initiation. Small ribosomal subunit binds to mRNA at the start codon, AUG. The first RNA, carrying methionine, binds to the start codon. Large ribosomal subunit joins to form the complete initiation complex. 2. Elongation. Ribosome moves along the mRNA 5-3 direction. RNA molecules bring amino acids to the ribosome. Each amino acid is added to the growing polypeptide chain via peptide bonds. The ribosome has 3 binding sites: A site, aminoacyl site, where new RNA enters; P site, peptidal site, where the growing peptide is held; E site, exit site, where RNA exits after delivering its amino acid. 3. Termination occurs when a stop codon is reached. A release factor binds, causing the ribosome to release the completed polypeptide. Ribosome subunits dissociate and are reused. Post-translational modifications. After translation, proteins often undergo folding into their functional 3D structure, assisted by chaperones. Cleavage of signal peptides. Addition of chemical groups, phosphate, methyl, acetyl, glycosyl groups. Transport to specific cellular locations. Genetic code characteristics. Triplet code. 3 nucleotides equals 1 codon. Universal, same in most organisms. Redundant, multiple codons for some amino acids. Unambiguous, each codon codes for one amino acid only. Not overlapping and continuous. Importance of translation. Converts genetic information into functional proteins. Determines cell structure, function, and regulation. Errors in translation can lead to non-functional or harmful proteins. Inhibitors of translation. Tetracycline blocks RNA binding. Prokaryotes. Chloramphenicol inhibits peptide bond formation. Prokaryotes. Cyclohexamide inhibits elongation. Eukaryotes. Puromycin causes premature chain termination. Comparison: transcription versus translation. Feature: transcription, translation, template, DNA, mRNA, product, RNA, protein, enzyme, RNA polymerase, ribosome, location, nucleus, eukaryotes, cytoplasm, direction 5-3, 5-3, along mRNA. Conclusion: translation is a highly accurate, regulated, and vital process in gene expression. It ensures that genetic messages stored in DNA are ultimately expressed as proteins, which control nearly every function in living organisms.